Hello everyone, Blake here to present the final video in our video series relating to the research section of your exam. In this video, we'll discuss graphical data, which includes forest plots, box and whisker plots, as well as potency versus efficacy graphs. Let's begin by talking about forest plots. Forest plots are graphical representations that are commonly used in meta-analyses and systematic reviews in order to easily display the results of multiple studies in one chart. The forest plot is used to identify a common statistic and display the results on a single axis. In order to understand the components of a forest plot, let's take a look at the example in this slide. The left column presents the names of the authors associated with each study. This provides a quick reference for the origins of the data. The x-axis of a forest plot is typically used to represent the measure of effect, such as the risk ratio, odds ratio, standardized mean difference, etc. In this particular example, we can see that the x-axis represents the odds ratio. The rightmost column contains the statistic, which is the point estimate and associated confidence interval. This offers insights into the precision of the individual estimates. Now at first sight, this might all seem a bit tricky, so to understand it well, let's go ahead and remove everything from the graph and study each component individually. Here we're left with the x-axis. The horizontal line typically shows estimates of effect sizes like odds ratio, risk ratio, standardized mean difference, etc. The vertical line is known as the line of null effect. It represents the point at which there is no difference or no association between the compared groups or variables. Now, if the confidence interval of an effect size estimate crosses or touches the line of null effect, it suggests that the observed effect may not be statistically significant at a given level, such as p is less than 0.05. Conversely, if the confidence interval does not cross or touch the line of null effect, it indicates that the observed effect is statistically significant. So now you might be asking yourself, how exactly are the confidence intervals presented? Well, each box, along with its horizontal line, corresponds to a study. The position of the box relative to the x-axis represents the point estimate. The size of the box corresponds to the study's sample size. The horizontal line indicates the 95% confidence interval of the study, with each end of the line representing the boundaries of the confidence interval. The shorter the line, the more precise the study. Conversely, the larger the line, the greater the uncertainty or variability in the point estimate. As we can see, this pink study represents a large study resulting in a narrower confidence interval. Conversely, this yellow study represents a smaller study with a much wider confidence interval. Notice how the horizontal line of this yellow study crosses the line of null effect. This means that the results of this study are not statistically significant, or in other words, the p-value is greater than 0.05. One more thing worth mentioning relates to the positioning of the horizontal lines relative to the line of null effect. When they are more to the left, this means that the study favors the experimental intervention or treatment being studied. When they are more to the right, this means that the study favors the control group or no intervention. The diamond of a forest plot serves as a summary statistic that combines the results from all included studies to provide a single estimate of the effect size across the entire body of evidence. If we drew a line through the center of the diamond, it would represent the combined point estimate of the overall effect. The width of the diamond corresponds to the 95% confidence interval around the overall effect estimate. Wider diamonds indicate greater uncertainty in the estimate, while narrower diamonds suggest more precise estimates. Forest plots allow researchers to quickly assess the consistency and magnitude of effects across multiple studies. This is what makes them so useful in systematic reviews and meta-analyses. They are also particularly useful for identifying patterns, heterogeneity, outliers, and assessing the overall strength of evidence. Next, let's discuss box plots, which are also known as box and whisker plots. Box plots graphically demonstrate the locality, spread, and skewness of numerical data through their quartiles, where min represents the minimum number in the dataset, Q1 represents the first quartile, Q2 represents the median of the dataset. Recall that the median is the middle number in the dataset when the data is arranged in ascending or descending order. Q3 represents the third quartile, max represents the maximum number in the dataset, and interquartile range, or IQR, contains all of the data within the first and third quartile. Now, as we can see, in a box plot, 25% of values lie between the minimum and the first quartile, 25% of values lie between the first quartile and the median, 25% of values lie between the median and the third quartile, 25% of values lie between the third quartile and the maximum, and 50% of values lie between the first and third quartile. Now, in order to calculate interquartile range, you take Q3, which is the median value in the upper half of the dataset, minus Q1, which is the median value in the lower half of the dataset. And finally, let's review a potency versus efficacy graph. The x-axis represents the logarithm drug dose concentration, while the y-axis represents the response. Now here we have two curves, one for drug A and one for drug B. In order to achieve the same response, we need a much higher concentration of drug B compared to drug A. This also means that we need a much lower concentration of drug A in order to achieve the same response compared to drug B, making drug A more potent. Now, at a certain concentration, as shown in the graph, drug B is able to elicit a greater response compared to drug A. This means that drug B has a higher efficacy compared to drug A. Drug A's response stagnates after a certain concentration, but drug B is able to keep increasing in response. Alright everyone, that's going to do it for the content covered in this video, so let's go ahead and practice what we've learned with an example question. What does the rightmost column of a forest plot represent? We have option A, study, B, subgroup of data, C, size of study, D, statistic, and E, spread. And the correct answer for this question is option D, statistic. So remember that the rightmost column represents the statistic that the studies being profiled show. This could either be a relative statistic, like an odds ratio or relative risk, or the statistics being used might be an absolute one, such as an absolute risk reduction or standardized mean difference. So, in summary, in today's video, we learned how to read and interpret forest plots, which are extremely useful for systematic reviews and meta-analyses. We also reviewed box plots and how to calculate interquartile range, and then we finished off with potency versus efficacy graphs. I really hope this was a good review over these concepts, and keep on studying hard. Good luck, everyone.